Good afternoon and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. Sarah Yip will present Toward Brain-Based Prediction of Recovery, How Neuroimaging Can Help Combat the Substance Use Epidemic. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 195 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the Foundation to fund creative and impactful, basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has had such great impact is because we do not limit our focus to one illness or condition. Since 1987, the Foundation has awarded over $461 million to fund more than 6,500 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorder, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Sarah Yip. Dr. Yip is an associate professor of psychiatry at Yale School of Medicine and the director of the Yale Imaging and Psychopharmacology Lab. She was a 2018 Young Investigator. Our webinar will begin with Dr. Yip's presentation, which will then be followed by Q&A. To submit your questions, please use the question tab on the control panel. Feel free to submit the questions at any time. Following the presentation, I'll ask as many as is possible in the time allotted. And now, I'm pleased to introduce Dr. Yip. Sarah, the floor is yours.
Thank you so much. I just had to unmute myself. Here we go. Hopefully you can see my slides.
Yes. Mm-hmm.
Well, thank you so much for the invitation to present. This is just, a massive honor. So I'm really excited to share my work with you all today. Okay, great. So I imagine that this audience is aware of the current substance use epidemic in this country, but I always think it's really important to mention. So this color map is showing population level overdose deaths per 100,000 individuals in the United States back in 2014. And as we know, these rates have really continued to rise. So for example, annual opioid associated fatalities in the United States exceed those caused by firearms and motor vehicles combined. And while it's been much less publicized, there really has been this concurrent rise in cocaine and stimulant associated fatalities. So this graph is showing the number of overdose deaths involving stimulants, so drugs such as cocaine and methamphetamine, over the past several years. And as you can see, there's been this dramatic rise since 1999 where the graph starts. And the graph is also breaking down stimulant overdose deaths as a function of deaths that also involved opioid use, as shown in the yellow line, versus those that didn't have any opioid involvement, as shown by the light blue line. So this is really highlighting that not only have stimulant overdoses also been increasing over the past 10 years, but that specifically overdoses involving combined use of both stimulants and opioids together have been increasing. Therefore, improved strategies to combat the current substance use epidemic really are urgently needed, which is the motivation for my lab's neuroimaging research. I do often get asked why we would even use neuroimaging to try to predict a treatment outcome in the first place. After all, we're pretty fortunate in the field of addiction. We have a large number of really excellent evidence-based treatments that are very effective for some individuals. However, we also have a lot of between-patient heterogeneity, which unfortunately means that the overall efficacy of any given treatment tends to be highly variable across individuals. And given that heterogeneity, it's perhaps not surprising that even gold standard treatment attempts tend to have high rates of relapse, and that most individuals with a substance use disorder go through multiple failed treatment attempts. In other words, when it comes to addiction treatment, one treatment doesn't fit all. In addition, relapse rates following treatment remain high, and for some substances, this really is a critical vulnerability period for overdose-associated death. And then finally, that problem is further exacerbated by the fact that what I like to refer to as sort of traditional clinical variables, so things like baseline severity, don't actually tend to be all that helpful for predicting treatment response or relapse rates, meaning that we have these unexplained sources of heterogeneity that are influencing our treatment outcomes. So within this context, a number of research groups, including my own, have turned to neuroimaging measures to try to identify brain-based predictors of treatment response to see if we can uncover hidden sources of variance that are related to individual differences in treatment outcomes. And over the past 10 to 15 years, there have been a number of proof of concept studies linking brain function and structure to treatment outcomes and addictions. So for example, work by our group and others indicates that individual differences in pre-treatment reward-related activations are prospectively linked to within treatment abstinence, that changes in functional neural circuitry may co-occur with reductions in substance use, and that individual differences in functional connectivity relate to both current cocaine use and future relapse to cocaine. So collectively, these and other studies support this overarching hypothesis that individual differences in brain function are indeed somehow linked to differences in treatment outcomes. However, this work, including my own, has some limitations. The first of which is that back when my group began using neuroimaging to study opioid use disorder in 2016, there simply weren't that many fMRI studies specifically focusing on treatment outcomes in for opioid use disorder versus other addictions. Rather, the majority of neuroimaging work for many years had focused on other drugs of abuse, such as cocaine and other stimulants. So in other words, for many years, the science was still catching up with the opioid epidemic. It takes a long time to collect data. So we have more data now, so this is starting to change, which is very exciting. The second limitation at that time when we started this work was that most prior fMRI studies of addictions had used methods that relied on prospective associations, in that they were sort of single time point designs that then sought to relate in a post hoc manner to treatment outcomes. So these weren't studies that were truly focused on prediction of a clinical outcome. So I'll talk about both limitations a bit more, but to begin to address this first limitation, back in 2016, my group undertook a systematic literature review of the existing fMRI data that was specifically related to opioid use disorder versus other addictions, which we published back in 2019. I won't spend too long on the findings from that review here because, as I said, it was published a number of years ago. But I do just briefly want to focus on what our review found when we looked at findings from studies in which they used fMRI to assess patterns of brain responses to drug images. So these are studies where someone is lying in the MRI scanner and being shown images of drug-related stimuli. And so what we have here is what we call a sagittal view of the brain. So you're viewing the brain from the side. And on the left, we have different brain regions color-coded. And then on the right, we have the same brain regions color-coded based on whether or not that region was increased in red or decreased in blue in response to drug cues among individuals with opioid use disorder. And so what you can see is that what we found was that data combined across all of these prior studies in opioid use disorder indicated that individuals with this addiction exhibited heightened neural responses to drug cues within brain regions involved in things like decision-making, such as the prefrontal cortex, and also within reward-related brain regions, such as the ventral striatum, really highlighting that this is a disorder that involves the brain and that there's a measurable brain-based difference between individuals with and without opioid use disorder. What we also found was emerging evidence across studies that this increased brain response to drug cues was in fact reduced by medications for opioid use disorder, as well as following prolonged periods of drug abstinence, thus suggesting that not only is opioid use disorder, at least in part, a brain-based disorder, but also that treatment for opioid addiction may involve changes in the brain. So again, this is excellent proof of concept data that aspects of opioid use disorder treatment may relate to individual differences in brain function. However, almost all of this research at the time had been done, as I mentioned, using single time point designs, where researchers were post hoc comparing individuals who did or did not do well in treatment, as opposed to scanning people at the start of treatment and then looking at predictors of change or even at sort of neural trajectories by scanning at multiple time points. Which brings me back to that second limitation I mentioned a couple of slides ago, which was that, again, most prior studies had been this single time point design. So therefore, my lab became very interested in trying to develop machine learning models of treatment outcomes, where we were scanning people early in treatment and trying to predict how well they would do over the course of time after the scan. This is my little picture of machine learning, by the way. It's a robot reading a book. So why are we using machine learning for this kind of research? Well, it turns out that in psychiatry and other research fields, the term predicts is often misused, and it's often used to refer to findings garnered from approaches using methods such as correlation or regression. And the problem with using this term predicts in the context of correlation or regression is that these approaches often have the tendency to overfit the data, limiting likely replication in external samples, when in fact, a true prediction really requires application of a model to novel data. Machine learning approaches seek to reduce overfitting by a creation of a predictive model in a data-driven manner, using a training data set and then application of that model to an independent test data set, which is used for model validation. And that distinction is really important because whereas the goal of traditional statistical approaches is to explain the relationship between two variables, the aim of machine learning approaches is to generate predictions in novel data. And I would argue that this type of approach really is critical for the eventual translation of research findings into clinical settings, which is a primary challenge of modern psychiatry. In addition, if used correctly, I would argue that these approaches can also be used for neurobiological discovery, which for me is critical, because in my opinion, in addition to generating predictions in novel data, one of the main goals of brain-based clinical modeling should really be elucidation of mechanism, because we know that even highly predictive models often can do little to advance mechanistic understanding. So throughout this talk, I'm really going to be trying to focus on maximizing neurobiological discovery within the context of predictive modeling or machine learning. In order to maximize such discovery, we use data-driven whole brain methods. The reason for this is that given that recovery from addiction involves complex interactions across clinical and biological domains, we think it's likely that achievement of abstinence also involves distributed processes from multiple brain regions. So therefore, all of the data I'm going to be presenting today is using a whole brain connectivity-based approach in which we're focusing on patterns of functional connectivity across the entire brain. So the specific method I'm going to be focusing on throughout the talk is referred to as connectome-based predictive modeling. And unlike some other machine learning approaches that require a priori specification of networks or specific brain regions, this is a whole brain approach. And therefore, it's both a predictive tool as well as a method of identifying networks. And prior to the data I'll be presenting today, this approach had been well validated and had been previously used to generate robust predictive models of IQ and attention and other trait-like behaviors, but it had never been applied to predict a future behavior or clinical outcome. In case at this point in the talk you're wondering what is a connectome, this term is just used to refer to the network of elements and connections that form the human brain. So in using this term, we're moving away from more traditional approaches that focus on isolated activity within a constrained set of brain regions and moving toward a network-based approach that focuses on patterns of communication across brain regions. I'm going to do my best not to go into too much technical detail today, but just really briefly. Normally, when we do a traditional fMRI study, we put someone in the scanner and we ask them to perform a specific task, or we show them a series of stimuli so that we can measure changes in cerebral blood flow over time and use this to make inferences about brain activity. So specifically, we can take the pattern of activity that we see in a given region of the brain and compare it to the pattern of activity that we would expect to see based on what we know about blood flow in the brain and what we know about the timing of the task events. And if that matched the pattern of that we expected to see, we would infer that this is a sort of task-related activity. And we can go even further by comparing patterns of activity to different kinds of stimuli. So in this case, we could compare, activity to burgers versus sushi, and based on the data, we could, for example, perhaps identify parts of the brain that are engaged in response to cheeseburgers, but not to sushi, and we might conclude that a given brain region is in fact the cheeseburger part of the brain. In other words, when we do traditional fMRI analysis, we focus on the temporal course of neural activity within a given brain region in response to a stimulus. In contrast, when we're studying the connectome, we're not necessarily interested in how our signal relates to a specific external stimulus, but we're very interested in how correlated our signals are across different regions of the brain. In other words, we're interested in the temporal coherence between brain regions. So now we have signal over time for two different brain regions, and we can correlate these patterns of activity across the time course of the whole scan to get a single statistic that summarizes the temporal coherence between these brain regions. And if the correlation between a given pair of regions is high, we're assuming that they're in fact functionally connected. So this is sort of an extension of the classic neuroscience concept of what fires together wires together. Or to take a paraphrased version from the film Titanic, you jump, I jump. In other words, we're assuming that brain regions that show similar temporal dynamics are in fact involved in the same processes. And we can actually do this for every single voxel in the brain to create what's called a functional connectivity matrix. So this single matrix is now summarizing whole brain patterns of connectivity for all of the voxels in the brain for a single person. So all of the possible pairs of voxels. And so this is a very powerful approach. Again, this one connectome is now summarizing patterns of connectivity for a given individual at a given scan. And there's data to indicate that these connectomes are both relatively unique to the individual, but also the patterns of connectivity within the connectome for a given individual may vary as a function of task performance, or what we sometimes refer to as brain state. And it turns out that brain state is really important because the accuracy of predictive models generated from connectivity data is actually improved when the input is data collected during task performance as opposed to data collected during resting state. So these two graphs just show the performance of connectome-based predictive models of IQ that were generated in two different cohorts and using different types of fMRI data, as indicated on the x-axis. And what I want to point out here is that in both cohorts, the predictive accuracy of models built using resting state data is relatively low. So in both cases, the model is only able to account for about 4% of the variance in IQ. The second thing I want to highlight is that the predictive accuracy of models generated from different types of task data is also highly variable, with some tasks accounting for up to 12% of the variance in IQ and others accounting for less. So together, this is suggesting that specific brain state manipulations may be preferable for predicting a given behavior, which is an issue I'll be returning to throughout the talk. And that brings me to the first bit of data that I'm going to be presenting today, in which we tested for the first time the ability of this connectome-based approach to try to predict a real-world clinical outcome. In this case, we were trying to predict abstinence from different drugs of abuse among polysubstance users. The clinical rationale for this work really came from some ecological momentary assessment data from a few years ago. This was an electronic diary study of methadone-treated individuals who had both cocaine and opioid use disorders. And in this study, what they did was they asked participants to track changes in their mood and craving and to document their substance use within this sort of electronic diary framework. And what they found was a differential association between positive versus negative mood states and future substance use, such that cocaine use was most robustly associated with having been exposed to the drug or being in a positive mood state, suggesting links with impulsivity and reward. But that heroin craving was associated with increases in negative affect, suggesting possible links with emotion regulation or inhibitory control. And so based on this finding, that different mood states predict opioid versus cocaine use behaviors in the same individuals, we are interested to see whether different brain states might be more closely linked to opioid versus cocaine use. To test this, we recruited patients entering a 12-week treatment trial and scanned them at the start of treatment and then again following treatment. All of our participants were receiving methadone maintenance therapy for opioid use disorder, but were now entering treatment for cocaine use disorder. So these were polysubstance using individuals, just like in the behavioral study I just mentioned. And so using these data, we wanted to see if we could develop these brain-based models of different substance use behaviors. As mentioned, the machine learning method that we're using is referred to as connectome-based predictive modeling. We've now used this in multiple publications to predict different substance use treatment outcomes. I'm showing a few of them here on your right. And just to iterate, a key advantage of this connectome-based approach is that in addition to generating a predictive model, it also identifies the specific neural connections underlying those predictions. And given that the goal of our research is to identify brain mechanisms of addiction treatment, it really is this feature, this identification of individual connections, that's most central for addressing our current research questions and for improving understanding of the brain mechanisms of recovery. So this is quite a heavy method slide. So if you're not interested in the actual machine learning model, please feel free to space out during this slide. But I did want to include just a really brief overview for those who might be interested. So practically, when we take this approach, what we're doing is we're taking individual participant functional connectivity matrices or connectomes from a training data set, and we're relating them to a behavioral variable of interest. In this case, we're relating them to within treatment abstinence. And we do this using simple correlation to identify positive and negative predictive connections, or what we refer to as edges. So positive edges are connections for which increased connectivity predicts abstinence, whereas negative edges are connections for which decreased connectivity predicts abstinence. So this is our feature selection phase. We then create individual participant summary scores by summing the edge weights identified in our feature selection, so that each participant now just has two values. One is a positive summary score and the other is a negative summary score. We then create our brain behavior model by entering these summary scores into predictive models, assuming linear relationships. So this is a simple y equals mx plus b. And then finally, we apply this model to our testing data. So we take connectivity matrices from an independent data set, we extract connectivity values from the edges we identified in our training data set, sum them to create summary scores for our new participants, and enter those values into our brain behavior model to generate individual predictions of within treatment abstinence. And finally, although it's not shown here, we evaluate the performance of our model by comparing those predicted values with the actual within treatment abstinence values for those individuals. We can compare the actual and predicted values a couple of different ways, but really the simplest is rank correlation, which is what I'll be using throughout this talk. Okay, that's the end of the technical bit. So if you spaced out, you can start paying attention again now. So for this study, we had these two different sets of connectomes. One set was summarizing, one set of connectomes was generated using data collected during performance of an inhibitory control task, and the other was set of connectomes was computed using data collected during performance of a reward task. And so we, computed these connectomes and entered them into our machine learning model along with our biological measure of abstinence, which we define based on the results of twice weekly urine testing over 3 months. So for this work, we have a dimensional biologically verified measure of abstinence. And then as a reminder, based on this self-report data showing that different mood states predicted decisions to use cocaine versus opioids, we were hypothesizing that different brain states would be related to cocaine versus opioid use behaviors. So specifically, we anticipated that patterns of brain connectivity during a sort of inhibitory cognitive brain state would relate to opioid abstinence, but that patterns of connectivity during a reward brain state would relate to cocaine use. And that was actually exactly what we found, which was pretty cool because that doesn't often happen. So here on the y-axis, we have an index of model performance, or the amount of variance in substance use that our brain-based model was able to account for. And what we can see is a double dissociation, such that on the one hand, our brain behavior model is successful in predicting opioid abstinence over 3 months when we use this cognitive task data, but not when we use our reward task data. And on the other hand, our brain behavior model is successful in predicting cocaine use when we use reward task data, but not when we use cognitive data. So these data are suggesting that not only are different mood states specific for opioid use versus cocaine use, but that different brain states may also be specific for predicting different substance use behaviors. One huge challenge of predictive modeling is, of course, whether or not the models that you create will replicate out of sample, which is an issue I brought up at the beginning of the talk. So this issue of generalizability. And we're in the process of collecting data to test the relevance of the opioid network in a separate external sample, which is really best practice. So we haven't done that yet, but we have replicated the cocaine findings in 2 external samples. So we do think these effects really are real. That said, as you can see from the graph, even the best performing models are only able to account for somewhere between like 10 and 20% of the variance in substance use outcomes. And that amount really is comparable to the amount of variance that models of trait behaviors such as IQ typically account for. Whereas here we're predicting something more complex, we're predicting a real world clinical outcome. So this isn't actually a particularly poor result. It's just that, it's not yet clinically actionable. And of course, even if we could explain much more, so even if we could explain, 50% of the variance, that's still not clinically actionable in terms of day-to-day decision making. And I think that's why it's really important to think of these sort of brain-based prediction approaches as ways of identifying neurobiological mechanism. These mechanisms are complex, so networks identified using this type of a connect-and-wide approach typically are distributed across multiple adjacent and non-adjacent brain regions, consistent with what we know about the complexity of psychiatric illness. That was certainly the case here. So this is just a visualization of the cocaine network separated out into positive and negative predictive connections. So as a reminder, positive connections are connections for which increased connectivity is a positive predictor of abstinence, whereas negative connections are connections for which decreased connectivity is a positive predictor of abstinence. And while these networks are certainly complex, it's important to point out that actually the combined number of connections for the cocaine network that I'm showing here actually comprises less than 2% of the possible connections that we could have identified using this approach. So in other words, despite the visual complexity here, these are actually quite specific connections. The opioid network was about twice as large, so it had about 1,000 connections, but hardly any of these connections overlapped. So here we have the overlap between the cocaine and opioid networks. So out of a possible 500 potentially shared connections, only 8 overlapped, and on the left I'm showing you those. So these are edges for which consistent relationships existed, with the red and blue lines indicating shared connections for which either increased or decreased connectivity predicted abstinence to both drugs. But then on the right, I'm showing you connections for which opposing relationships existed. In other words, connections for which increased connectivity was a positive predictor of cocaine abstinence, but a negative predictor of opioid abstinence, or vice versa. And as you can see, there are more opposing than consistent connections. In other words, there was very little anatomical overlap, really demonstrating the ability of this type of machine learning approach to not only generate predictions and unseen data, but also to provide novel biological insight, which we can hopefully use to guide development of better treatments. One helpful way that we can begin to understand our complex abstinence networks is via their spatial overlap with what we sometimes refer to as sort of canonical neural networks. So these are so-called resting state networks that you may have heard of. So for example, things like the default mode network or the frontal parietal network. So here we just have matrices that are summarizing overlap with those canonical networks for the positive and negative cocaine abstinence networks separately. And for each matrix, the cells are representing the total number of connections within and between each network, with the darker colors indicating a greater number of connections. When we do this, we can see that the positive network is characterized by a large number of frontal parietal and medial frontal connections, as well as by a lot of salience and motor sensory connections, as indicated by these dark red boxes. And we can also see that the negative network includes a large number of salience, default mode, and medial frontal connections, as indicated by the dark blue boxes. And then these differences become even more apparent if we directly compare the number of connections within each network. So we can see that the positive network includes more frontal parietal to medial frontal connections, as well as more salience to subcortical to motor sensory connections. In contrast, the negative network includes more connections between the medial frontal network and salience and default mode connections. So based on these differences, we've generated a theoretical network-based model of cocaine abstinence. So our data are indicating that cocaine abstinence is positively predicted by increased connectivity within and between frontal parietal and medial frontal networks. As a reminder, these are systems involved in coordination of attention and executive control. So we think that these might contribute to within treatment abstinence via coordination of top-down processes necessary for treatment engagement. So things like acquisition of new skills and enhanced control over impulsive behaviors. Our data are further indicating that cocaine abstinence is positively predicted by increased connectivity within salience, subcortical, and motor sensory regions. And these are networks involved in coordination of salience encoding and rewards. So we think that these networks might support things like motivational processes relevant to treatment. So things like willingness to change and shoring up of non-drug reward processing and attending to alternate rewards. Finally, our data indicate that abstinence is further predicted by decreased connectivity between these two systems. And so based on prior connectivity work in cocaine use disorder, we think that appropriate separation between these two systems might relate to greater behavioral flexibility or decreased compulsivity, as might be required for behavior change during treatment. So really this model is building on prior models of addiction, emphasizing separation of frontal parietal and salience networks, but also now incorporating medial frontal, motor sensory, and subcortical networks to provide a theoretical framework for future research. And our hope is that by proposing this framework, we can encourage others to test these hypotheses in their own data. And we've also publicly shared all of the actual anatomical masks for these data and the code for our analyses. As already mentioned, when we turn to the anatomy of the opioid network, it's actually very different. And in fact, in some ways, it's much less consistent with classical models of addictions neuroscience. So we've again proposed a theoretical network model that summarizes key aspects of this network. So this figure just emphasizes that, as indicated by the red lines, opioid abstinence was associated with increased within network motor sensory connectivity and increased between network motor sensory and salience network connectivity and increased between network default mode and frontal parietal network connectivity. The model is also emphasizing that abstinence was further associated with decreased connectivity between the motor sensory network and default mode and medial frontal and frontal parietal networks, as indicated by those blue lines. And again, our hope is that by summarizing the data in this somewhat simplistic way, we can encourage others to test these theories in their own data sets to further guide neurobiological discovery. We've also tested the ability of the identified networks to predict abstinence following treatment. So for this analysis, we simply create summary scores by summing connectivity weights for the connections identified in our original analysis. So we take our abstinence networks and we use them as a mask to extract values from neuroimaging data acquired 3 months later at the end of treatment for cocaine use disorder. And we entered those summary scores into correlational analyses with percent days of abstinence during follow-up. And we find that, in fact, for both the opioid and cocaine networks, post-treatment network strength are, in fact, significantly correlated with post-treatment abstinence for each drug, respectively, thereby indicating relative consistency of this relationship over time. Further consistent with that, when we just compare pre versus post-treatment connectivity strengths, we didn't see any changes over time, raising the possibility that these effects are actually somewhat stable within individuals, which for me is really interesting and touches on an important point, which is that networks contributing to treatment response may be distinct from those that change with treatment or that differ from non-addicted individuals. So, for example, brain regions predictive of treatment responses in other disorders often have limited overlap with regions consistently found to differentiate patients from controls. Another possibility is that brain regions which predict treatment outcome may just be different from those that change with treatment. And so at first that can seem really counterintuitive. However, when you think about it in a clinical context, we know that factors that predict treatment response, such as motivation to change, are in fact can be distinct from those that change with treatment, such as acquisition of new skills. Therefore, the same may be true of neural networks. Further, it's also possible that changes within abstinence networks may just take time to emerge and therefore may only be detectable months after treatment. And so that theory is consistent with data demonstrating that abstinence rates continue to improve following some behavioral treatments for cocaine use disorder. Thus, it stands to reason that neural change might also take time to emerge. And so we've begun trying to explore that first possibility, or the possibility that our networks are not only predictive of future substance use, but that they also just may be altered relative to healthy control individuals and therefore perhaps linked to addiction pathophysiology more generally. So the theoretical basis for this work came from a nice paper from Hugh Garavan and colleagues from a couple of years ago, in which they proposed a few different scenarios for what prolonged abstinence might look like at the brain level. So here at the top, they proposed that one scenario could be that recovery from addiction could involve some sort of restoration of premorbid brain function. In the middle, they proposed an alternative hypothesis, which would be that prolonged recovery may in fact require some sort of hyperfunctionality of brain regions involved in abstinence maintenance to above levels observed in healthy controls. Or finally, a third option at the bottom is that individuals in recovery from addiction may continue to exhibit decreased function, thereby conferring vulnerability for relapse. So just to unpack that a little bit more, based on this model, if we were to compare our abstinence networks to those of healthy controls and found that abstinent individuals had similar levels of network strength relative to healthy controls, we might conclude that what we're seeing is a return to premorbid functioning. Alternatively, if we were to find that individuals who achieve abstinence have increased network strength relative to controls, we might conclude that what we're seeing is an elevation of brain function or a sort of hyper recovery. And finally, what we were to find is that abstinent individuals had decreased network strength relative to controls. We might interpret that as indicating a continued vulnerability for relapse. So to test these theories, we computed connectomes from identical task data from 38 age and sex-matched non-substance using control participants, and then compared that to our polysubstance using sample. So here I'm just showing you a binarization of the data I presented earlier. So we have the cocaine network, we have strength of the cocaine network for individuals who achieved some abstinence from cocaine during treatment on the left, and then network strength for individuals who did not achieve any abstinence on the right. So this is, again, just a binarization of what I presented earlier. What becomes interesting is when we add in our healthy controls and we find that non-substance using individuals are actually intermediary between treatment responders and non-responders, such that our treatment responders actually have increased network strength relative to controls, and that our non-responders have decreased network strength relative to controls. We see a similar pattern when we look at our opioid network. We find that our control group is, again, intermediary between responders and non-responders, with responders having increased network strength relative to controls, although the difference between controls and non-responders here is not significant. So for both the cocaine and opioid networks, we're seeing this pattern that treatment responders have greater network strength than control participants, again, consistent with this notion of a sort of elevation of function relative to controls, or hyperfunctionality. I have to say I really love this result. In some ways, it directly contradicts the misconception that individuals with addictions have poor willpower or somehow weak, because really it's suggesting that recovery from addiction requires a sort of superhuman network strength to above the level observed in your average healthy control. And so some of our ongoing work is like focused on understanding how this effect might occur. So we're tracking network strengths over time during early treatment at a high temporal resolution. I'll talk about that a bit more at the end, but first I just want to highlight some of our other ongoing work, which is our developmental work. So in addition to studying brain mechanisms of recovery, my lab is also very interested in understanding brain-based risk factors. So in one recent example of this work, we sought to identify predictive neuromarkers of alcohol use risk in a large sample of youth. And in doing so, we were again interested in understanding the effects of brain state on prediction. And also for this sample, we were interested in exploring the effects of biological sex, which isn't something we've been well powered to do in our smaller clinical data sets, but we nonetheless think is very important. So for this work, we used data from the Imaging Consortium. This is a large multi-site longitudinal study of adolescent development that was conducted in Europe and began a few years ago. Individuals in this cohort were first scanned at age 14 and then again at age 19. During scanning, they performed a reward task and also an inhibitory control task. So these are very similar fMRI data to our clinical data sets. And then at both time points, all of the participants in this cohort also completed self-report assessments of current alcohol use severity using the Alcohol Use Disorders Identification Test, which is this widely used and well-validated clinical screening tool for alcohol use disorder that was developed by the World Health Organization. So this data set really is ideally suited to answer these critical developmental questions about neural predictors of alcohol use. Here we have the distributions of alcohol use severity scores at ages 14 and 19. With age 14 at the top. When we look at the distribution of audit scores at age 14, we can see a very limited range of alcohol use behaviors, with most scores hovering around 0, consistent with the young age of participants. So this is a good thing. Our 14-year-olds are not drinking a lot. However, at 5-year follow-up, we see a much more varied distribution of audit scores. And then for reference, I'm just going to overlay the different audit score cutoffs here. So you can see that a score of 0 indicates no alcohol use. A score of 1 to 7 indicates low risk use. A score of 8 to 14 indicates hazardous use or hazardous or risky alcohol use. And then a score of 15 or higher likely indicates an alcohol use disorder. So you can see that at age 19, most of the adolescents in this European cohort are falling into either this low risk or hazardous risk group. So most of them are drinking, and a handful are even meeting criteria for likely alcohol use disorder. So again, given these distributions, all of our analyses for this data set focused on predicting alcohol use at age 19, since there wasn't a lot of use at age 14. But we're doing this using the neuroimaging data acquired at both age 14 and age 19. So using the age 14 data, we're trying to predict future alcohol use at age 19. And then using the fMRI data acquired at age 19, we're trying to predict current alcohol use severity. And so using these fMRI data, we've again just generated connectivity matrices, which we enter into our connectome-based model to try to predict alcohol use severity. And then, of course, we're mapping the features identified by the predictive model back to neuroanatomy to provide elucidation of mechanism. And for those of you who are interested in some of the more technical aspects, we're running our models in a leave one site out manner. So we're generating our model in 7 of the 8 sites and then testing it in the 8th site in an iterative manner. And this really is the gold standard of this sort of approach for multi-site data. So here we have the initial model results when we're trying to predict future alcohol use severity using the neuroimaging data acquired at age 14. So we're taking imaging data from age 14 and trying to predict severity at age 19. And as you can see here, both the sex combined model and the female only models were successful in predicting future alcohol use, whereas the male only models were not at age 14. And in fact, we can also see that the female only models were successful in predicting alcohol use across both brain states or task types. So on the y-axis, you have the strength of the identified brain behavior relationship as determined using our leave one site out cross-validation. And as you can see for females, this is approaching a correlation coefficient of 0.3 for the correspondence between actual and predicted values, which actually is on the higher end of what most recent work suggests one should expect for the size of adequately powered and cross-validated brain behavior associations. So this is a decent model result, even though it's still not, enough for any sort of clinical decision making. When we try to predict alcohol use severity using the data acquired at age 19, we again have good predictive accuracy in females for both reward and inhibitory control brain states. But in fact, we only achieve a stable model in males when we use inhibitory control task data, indicating some sort of brain state specificity in the males or indicating that inhibitory control processes may be more relevant than reward-related processes in understanding alcohol use risk in male adolescents. In the interest of time, I'm not going to go too much into the anatomy of these specific networks today, though I do encourage you to check out the paper. And also, again, I want to point out that we've shared the actual anatomical masks for these and the codes so that people can explore these networks in their own data. But briefly, when we look at anatomical overlap across models, not surprisingly, we see much higher consistency across tasks, or brain states and time points across female-only models relative to male-only models. So these matrices are summarizing feature overlap between models developed using reward and inhibitory control tasks in females and males separately, and using connectivity data acquired at baseline, age 14, and then at 5-year follow-up at age 19. And then feature overlap for the positive predictive network connections is shown in red, yellow at your left, and overlap for the negative predictive network is shown in blue, green on your right. And what we can see here is that there's relatively high feature overlap for female-only models derived from the 2 different task types, or high brain state overlap in females, but much less brain state overlap in males. We also see that for females, there's relatively high feature overlap between models developed using data collected at ages 14 and 19, suggesting some sort of relative stability of network features across time or high temporal overlap in females. Whereas in comparison, we see much less feature overlap among the male-only models, which makes sense because the accuracy of the male-only models was both temporally specific and brain state specific. In terms of the actual anatomy, we can see that for females, the anatomy of positive and negative predictive networks identified at age 14 is relatively distributed. So here on the left, we have these matrices that are, again, just summarizing anatomical overlap between canonical neural networks and our alcohol neuromarkers, again, just for positive and negative predictors separately. So for each matrix, the cells are representing the total number of connections within and between each network, with darker colors indicating a greater number of connections. And then on the right, we have virtual lesioning results for each canonical network. So for these analyses, what we're doing is we're iteratively knocking out connections for 9 out of 10 of our canonical networks, and we're rerunning our model to determine the impact of the individual remaining network on model accuracy by itself. And when we do this, we can see that for the female network predicting future alcohol use, the networks that are contributing the most to prediction include medial frontal, frontal parietal, motor, salience, and cerebellar networks. We again see a similar pattern for the anatomy of networks identified at age 19 as predictive of current use for females. Although at this time point, the motor and cerebellar network features are now emerging above and beyond the prefrontal cortical connections in terms of predictive weight. And again, that pattern appears somewhat consistent across brain state in the females. Whereas when we interrogate the anatomy of the male-only network, we also see strong predictive power for motor and cerebellar networks, but even less involvement of cortical networks. So for example, very little medial frontal involvement. I do want to point out that we have, of course, also tried combining brain states. So when we do this, we find that for females, predictive accuracy is relatively unchanged when we use data acquired during both reward and inhibitory task states versus when we use only a single brain state, really indicating that in this instance, the information being captured by the connectivity data is actually somewhat similar in terms of its relevance to alcohol use in female adolescents across different brain states. In contrast, when we combine tasks in males, we actually get a decrease in model performance to below what we can achieve when we use inhibitory control data by itself, really indicating that not all brain states are created equal. We've also done a bunch of sensitivity analyses to test the robustness of our models. When we do this, we find that our model accuracies are robust and the features are really relatively unchanged, and we control for common confounds such as residual motion during scanning, or in the instance of predicting current alcohol use at age 19, if we control for baseline use at age 14, our models still predict and the features are relatively unchanged. Importantly, this is also the case when we control for common sources of phenotypic variance, such as things like trait impulsivity and trait neuroticism, really indicating relative specificity of the identified connections for predicting alcohol misuse specifically. As a further test of the robustness of our effects, we again sought to determine whether the identified neural networks would relate to alcohol use risk in an entirely separate sample. These were youth who were scanned completely independent from the European cohort. They were scanned in a different country. They were scanned in the United States. They were scanned on a different type of scanner using different image acquisition parameters during performance of a different type of inhibitory control task. And they even had different alcohol use risk assessments. But despite those many differences, the connections that we identified in our European cohort subserving alcohol use risk indeed replicated to predict risky alcohol use in this totally external test sample. For me, really demonstrating the robustness of this approach for generating meaningful brain behavior associations that we think will sort of generalize across multiple cohorts. I don't have time to go into all of our work today, but other ongoing developmental work in the lab includes identification of a problem cannabis network. This is work led by Sarah Lichtenstein, a super talented assistant professor in my lab. And so for this work, what she did was she used the same college sample that I showed you in the previous slide. So this is a sample of 118 adolescents in Connecticut. She used this non-clinical sample to identify a problem cannabis network. So a network that was associated with problem level cannabis use in this non-clinical sample. And then she went on to replicate that network back in the European cohort, which is also, of course, a non-clinical sample. So she's identified and replicated this network in a group of, again, non-clinical, so subsyndromal individuals that use cannabis. And then finally, she took that validated network to a separate, much smaller sample of individuals scanned prior to treatment for cannabis use disorder. And what she found was that the same network was linked to both baseline addiction severity and also to subsequent abstinence during treatment in this clinical cohort. So this is really an example of how we're trying to start to think about leveraging smaller clinical data sets using larger non-clinical data sets, which I think is a really important direction for future work because it's really hard to get large samples of clinical neuroimaging data. That's most of the data that I'm going to present today, but I think it really demonstrates both the ability of our approach to generate specific externally valid predictions, but also to provide neurobiological insight. So hopefully what I've demonstrated today is that networks identified using these whole connectome-based approaches are in fact clinically relevant. That is, they're able to predict real-world treatment responses. These networks are also externally valid in that they're able to generalize to predict specific behaviors in novel samples and individuals. In addition, these networks are robust to common sources of variance, so they predict even after controlling for severity or treatment assignment or medication status in the case of our abstinence networks, and they also predict across sites and after controlling for related phenotypes in the case of our alcohol use networks. Finally, despite their complexity, these networks are in fact biologically meaningful in that they're composed of specific connections observing specific behaviors. And therefore, this approach can also be used for robust identification of biological mechanism. Within that context, I think it's really important to consider model findings across multiple levels of interpretation, with the most basic level being the connections themselves and the most abstract level being the overarching neurobiological model, which can be used to guide treatment development and act as a basis for further testing. One simple method for developing these models and for determining the significance of different brain features is to rerun the model, including or excluding specific features. This is that virtual lesioning approach that I showed you with the alcohol networks. And we do this to determine which features are necessary for optimal model performance. So for example, this would let us answer the question, do we actually need to include amygdala connectivity in order to have a successful model? Similarly, you can rerun the model only including selected features, and that will enable determination of the specific weight of each feature alone. So for example, the question, what is the predictive ability of amygdala connectivity by itself? These are very simple steps, but people often skip over them. So this is just a reminder that identification of biological mechanism should also be a primary goal of brain-based prediction. If you're interested in learning more about this kind of approach for neurobiological discovery, I recommend checking out 2020's special issue of Biological Psychiatry, CNNI, which was dedicated to data-driven approaches. That included our review paper that covers recommendations for best practices and cross-validated biomarker research within the context of addictions, though many of the issues we cover apply to clinical predictive modeling more generally. In the interest of time, I won't go through all of those recommendations here, but I do want to highlight a few key points. One of which is careful consideration of the window of assessment. So scanning prior to any clinical intervention is often considered optimal, but I think it's really important to acknowledge that scanning during this time can introduce unnecessary confounds, such as effects of acute intoxication or withdrawal in the case of addiction, or of mood or psychosis state in the case of other disorders. In addition, scanning prior to clinical intervention may not always be feasible unless you're delaying treatment initiation, which can, of course, place unnecessary burden on the patient. So you don't necessarily have to scan prior to treatment. You just have to be clear about what the limitations are of the time point at which you're scanning. Hopefully, I've already also emphasized the importance of conducting post-hoc testing to provide elucidation of mechanism, because without this, findings from models, even with high accuracy, can do little to advance our understanding of the underlying neurobiology, which is essential for informing novel treatment development, and which really, in my opinion, should be the ultimate goal of clinical neuroimaging. And then finally, this arrow here is meant to indicate that this is an iterative process. We're still in the stage of identifying basic mechanisms of effective treatments. And so this sort of predictive approach needs to be thought of in that context, as opposed to as something we're ready to use to guide clinical day-to-day decision-making yet. Oh, whoops. And then in terms of other future directions, we have a lot, but just briefly, one example I wanted to mention of ongoing work is our work trying to understand the sort of longitudinal relevance of brain state and longitudinal trajectories of our neuromarkers over time. So here we're really trying to drill down into the longitudinal relevance of brain state for understanding clinical phenomena by sort of expanding our temporal resolution. So we're doing this via weekly scanning of individuals in early methadone treatment over 2 months. And of course, while we're doing this, we're also tracking their substance use during this time. So far, we've completed scanning from 10 individuals over 68 sessions, and we have additional funding to scan an additional 10 people over 60 sessions. So ultimately, we'll have over 120 individual scanning sessions from 20 people early in methadone treatment. So this is big data in a very different sense. You know, most conceptions of big data think about scanning a lot of people, and this is scanning, you know, a very small number of people a large number of times to try to look at how the brain is changing during early treatment. We're still collecting the data, but, you know, based on our prior data, you know, I just wanted to present a sort of theoretical example of what we might find. So based on our prior work with the abstinence network, what we, if we were to scan someone over time and to plot the trajectory of someone in recovery who was able to reduce opioid use during treatment, as shown by this gray line, or I think that's green, sorry, as shown by this green line, we might see this sort of initially a low abstinence network connectivity strength followed by an upward trajectory such that abstinence network strength would increase as the individual continued to do well in treatment. Whereas if we were to plot the trajectory of someone who didn't respond to treatment and instead continued to use throughout early treatment, we might expect a relatively flat trajectory with the overall network strength again being somewhat low at the start, but then remaining so throughout treatment. And then finally, if we were to look at the abstinence network strength for someone with intermittent opioid use, we might anticipate a much more dynamic trajectory that would track with use. So within this context, one treatment implication might be to design some sort of intervention that could be implemented here to prevent a return to opioid use. This is still theoretical in very early days. It takes a lot of time to collect this sort of data, but this is an example of the type of approach that we're really excited about. This is really a personalized medicine approach. If you want to learn more, I'll refer you to some of the reviews I mentioned earlier and also to a 10 simple rules for predictive modeling paper that we published in Neuroimage back in 2019. And also to a special issue of Biological Psychiatry that I guest edited with Anna Canova last year, which covers a lot of the issues I mentioned today, as well as many issues that I didn't cover. I'll end there, but I want to really thank everyone at the Brain and Behavior Research Foundation for funding this work, all of my collaborators and all of the co-authors on the data, which I've highlighted at the top here, and of course, everyone in my lab and all of the various funders, and all of you for listening. Thank you.
Right. Well, Sarah, thank you for a presentation packed with a lot of important information. Really just very exciting work that you and the team are doing. You made the point, and I think it's an important one that I want to emphasize, that this really shows the fact that chemical dependency is a brain disorder. It's an illness. It's not neural weakness, etc. I'd like you to comment on that.
Yeah, thank you so much. Yeah, I mean, again, I'm so excited about these data suggesting that you sort of need this like hyper functionality of your abstinence network to achieve abstinence. So this is directly in contrast to this sort of old-fashioned conception that there's some sort of weakness involved in addiction. And I think we for many years many psychiatric disorders are recognized as being brain-based disorders, and addiction, I think it's taken the science to catch up. I think people have not thought of it as much of a brain-based disorder as other disorders for a long time, and that's really fundamentally changed with all the research in the past 20 to 30 years. I mean, and I think the public is starting to understand that as well. I think general conceptions are that it definitely is a brain-based disorder. It's not just a brain-based disorder, just like things like depression, aren't only a matter of medication. Individuals with opioid use disorder or other addictions also need social support and housing and all sorts of other things, but there certainly is a brain basis as well.
Yeah, thank you. Where do you see this going? What if you were going to predict where this work will be in 10 years from now, 5 years from now, 20 years from now? What's your prediction?
Great question. I mean, I think it's really going to be this sort of personalized, what we call like personalized medicine, where we're, scanning an individual over time. So this work has been done increasingly in sort of basic human neuroscience. So there's a study from a number of years ago that I just love, where it's literally three researchers. It's a PI and like two of their postdocs, and they scan themselves every single day for a couple weeks, and then they each had a cast put on one of their arms, and then they kept getting themselves scanned, and then they had the cast taken off, and they kept getting themselves scanned. And what you see within days is this reorganization of the motor cortex to favor the non-dominant hand, or the non-immobilized hand that occurs after putting the cast on, and that reverses after taking the cast off. And for me, this is such an elegant brain-based example of how a basic clinical intervention might work. And I think that we have nowhere near nothing approaching that kind of understanding for psychiatric disorders. So this notion of sort of denser sampling over time, I think, is really important. And that's for sort of understanding basic mechanism. In terms of sort of the more clinical aspect and how we can apply this type of thing, I think that, again, it's important to consider the brain as incredibly important, but also one of many predictors. So I think studies, where you're incorporating fMRI scanning, but you're also, characterizing people across multiple different levels of behavior, and perhaps also incorporating, other biological measures as well, and then generating predictive models to try to guide treatment selection would be where the field is going.
Great, thank you. Well, we're gonna obviously want to have you come back down the road to share with us where this goes.
Thank you so much.
Yes, thank you. And I want to thank everybody for joining us today for this outstanding presentation by Dr. Yip. I hope that you can join us later this month for the Mental Health Research Symposium. It's on Friday, October 25th, where BBRF's 2024 Outstanding Achievement Prize winners will present their latest cutting-edge research. To register to join either in person or virtually, please go to our website. I also ask that people consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This means that if you donate $1 for research, that dollar goes directly to the scientists. To make a gift, please visit bbrfoundation.org or call us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it, please visit the events and webinar page on our website. And please join us again on November 12th at 2 p.m. Eastern Time, when Dr. Bridget Callahan, an assistant professor in the Department of Psychology at the University of California, Los Angeles, and director of the Brain and Body Lab in the Department of Psychology, will present Intergenerational Effects of Adversity on Mind-Body Health, Pathways Through the Gut-Brain Axis. Thank you, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Thank you.